Hello, everyone, and welcome to America's finest city. This is the backyard where I grew up, so I'm visiting some family in San Diego. I thought you'd like to see my backyard, and if you can't quite see, this is definitely the backyard that you would expect a future biology teacher to spend a lot of time in growing up. We got a large variety of flowers, fruit trees. I got a grapevine, got a lemon tree, lots of biology around me right now. So I hope you enjoy seeing the backyard of the house I grew up in. Let's jump into our continued discussion of our unit ion cell communication. I'm going to continue telling you the most important things to know from the textbook, so you don't have to read the whole textbook. This chapter is very short, so if you have time to read it, great. But I'm going to tell you the most important things so that you don't have to go through it if you don't want to. So last time we talked about different types of cell communication among different types of cells. You should know how yeast cells communicate in order to find mates. You should know how bacteria communicate in order to find food and good environments. You should know the difference between short and long distance communication. You should know how plants communicate using the plasma modesmata. So those are kind of our big topics. We did a few more things than that, but now we're on to signal transduction. So signal transductions have 3 steps. So I'll make 3 different videos for you about each step. The first step is reception. And there's actually quite a bit to know about cell reception. All really started with the work of a team of scientists headed by a scientist named Sunderland at Vanderbilt University, a great research university for those of you who are still considering where to apply to college. The work found out that the reception of the hormone epinephrine was not able to cause the chemical signaling, was not able to cause the effect or the response of the cell in the lab. So we would say in vitro. So in real cells, this is happening, but somehow if you just put glycogen and epinephrine and the things that would in a test tube, let me read the exact things they put in the test tube. So they found out that epinephrine and glycogen and the other normal enzymes needed were put into a test tube. No glycogen breakdown occurred. So what was going on? Well, what they proposed and what they hypothesized and what they eventually proved was that actually having a live cell was essential. And what we know now is the reason why that's essential is because of step 1 reception, that unless you have a live cell that has these receptor proteins, in this case of epinephrine, that the whole process doesn't work. So you could have everything else, but if you don't have the right thing to receive the epinephrine, the signal will not be transduced. Pretty interesting. Now, and that's the first type of signaling reception, and the most common that we see is that it's when a hormone or some sort of ligand, it could be any ligand, interacts with a receptor protein on the outside of a cell. And the most common one that you should know a little bit about are called G protein coupled receptors. These very nice receptors, and I'll see if I could show you a quick picture. If it doesn't look nice, I'll just edit this part out. Yeah, not a bad picture. They kind of look like this. Hopefully you can see. So what's cool about these proteins is, to go back to what we said, is they have lots of alpha helixes, which you can see. So it's a transmembrane protein. Those alpha helices help it span the membrane. And they have these really interesting turns that have specific sequences that allow them to bind to that specific ligand. Remember, a ligand by definition will bind only to a specific receptor. So even though epinephrine is released throughout the whole body and circulated by the circulatory system, sorry about that, I bumped the camera. Even though it will circulate the whole body, it will only interact with the cells that have the epinephrine receptor. And that is called signal reception. But that's not the only way it works. And you also should know that in addition to these G-coupled protein receptors that we bind to it and then activate the G protein by replacing GDP with GTP, and that causes other enzymatic activities, which we'll talk about next time. We can also have a receptor tyrosine kinase. I have a picture of that one for you. It looks like this. And this is another receptor that's in the membrane. Hopefully you can see that okay. And it's called a kinase because it leads to a phosphorylation cascade. So that's an important thing to remember about these. They have lots of tyrosines. That's why they're called tyrosine kinases. And those tyrosines are going to take phosphates from, in this case, there were 6 tyrosines. So they're going to take 6 phosphates from 6 ATPs. That's obviously a big energy investment. But with those 6 phosphates, from those 6 ATPs, it can now lead to a phosphorylation cascade. And that's going to lead to a whole lot of enzymatic activities. Unlike our ligand-specific receptors that we're talking about, those G protein coupled receptors are specific to one ligand. These can be used for a variety of different processes. And lastly, another one that's a receptor could be an ion channel receptor. And these can be ligand receptors, or they can actually be electrical, meaning when they receive an electrical signal in your nervous system, then that can cause them to open or close their ion channel. So all 3 of these are proteins. They're all proteins that are receiving some sort of ligand, that are receiving some sort of message, or receiving some sort of signal, and that's causing the transduction to occur. Pretty cool. But what happens if we don't have a receptor? Are we out of luck? Well, interestingly, occasionally we also have the situation, which is the one example you should know is testosterone, where there's not receptors for testosterone. And that might seem surprising, but the way your body has got around not having receptors is that testosterone is actually a steroid. And because it's a steroid, it is able to actually be hydrophobic enough, nonpolar enough to get through the cell membrane. So if we have a hormone that is hydrophobic enough so that it's able to go through the cell membrane because it's a steroid like testosterone, or it's something like nitric oxide, these are able to diffuse easily into the membrane. Then we don't need the receptors for them, and we can actually bind with them inside of the cell. So there are some exceptions to this reception binding, but if the hormone is not able to get into the cell, then there needs to be a receptor for it. If it is something like testosterone or nitric oxide or ethylene, these things are hydrophobic enough for them to get into the cell. So keep that in mind. Okay, so a couple of questions for you to think about as you study this section. These are, again, straight from your textbook, so I want to give credit to your good biology textbook. So first question is, nerve growth factor is a water-soluble signal molecule. Would you expect the receptor for NGF to be intracellular, like we were just talking about with testosterone, or in the plasma membrane? Question 2. What would be the effect if if a cell made defective receptor tyrosine kinase protein were unable to dimenterize? Okay, and when those become active, by the way, they become dimers. So I skipped over that, was in the picture, but when part of that activation, when they take the 6-phosphates, is they actually go from being separated, which their inactive form, to joining together. That's called dimerize. So you should know that. And lastly, how is ligand binding similar to the process of allosteric regulation in enzymes? So they want you to review allosteric regulation. There's a lot of similarities because with regulation, we also have ligands binding. So kind of wanting you to remember that what is this term ligand, and that it's not just used in cell communication, but in other things going on in the cell as well. One of the big similarities I'll tell you for that one is we know that chemical reactions are either irreversible or reversible. So these are both the same. I won't tell you which one. You should quiz yourself and know that already. Okay, that's it. So we talked about 3 different examples of reception. The first one, we'll start with the strange case, was the case of testosterone. In that case, it can it interacts with receptor proteins on the inside of the cell, and you should know why it's able to do that. The second case was our ion channels, which is very common in the nervous system. The third case was the receptor tyrosine kinases, and our fourth case was our G protein coupled receptors. I hope that helped you in your studying. If you have any questions, let me know. And that's it for today. One thing we should check before we go, though, is our unit guide. So let's check that real quick. So you get to check with me. Let's see if we forgot anything important. If we didn't, I'll just cut this part out of the video. Okay. College board. I have it saved. I hope you're all having a very wonderful break. I'm enjoying being in San Diego and catching up on sleep. Oh, my computer is about to die. Okay, let's go plug it in. I gotta go. My computer's gonna die, but have a good one, everyone.